Today we are going to discuss about the blood grouping and the compatibility. I have prepared two parts for this video. In the first part we'll be mainly discussing about the blood group systems, that is the ABO system and the RH system. And in the second part we'll be discussing about the compatibility of the various blood groups among one another. So the blood group or the blood type depends upon two aspects. See, whenever we have to tell the blood group of a person, we must know two things. The first is the presence or absence of antigen on the surface of RBC. And the second is presence or absence of antibodies in the plasma. So we need to know about two aspects. First is antigen and then is antibody. Antigen will be present on the surface of the RBC and the antibody will be present in the plasma. Plasma is the fluid in which all the cells are suspended. And the antigens can be either proteins, carbohydrates, glycoproteins or glycolipids based on the blood group system. It is not necessary that every time the antigen will be protein only. But in this case, in the ABO blood grouping system, mainly the antigens are proteins. But there are many other blood group systems about which we might not be knowing and that are not of very much significance also. In those systems, the antigens can be either carbohydrates, glycoproteins or glycolipids. So, a total of 35 human blood group systems are now recognized by the International Society of Blood Transfusion. This is the society that is responsible for studying all the blood grouping system. So they have till now discovered 35 different human blood group systems, but we are going to consider here mainly the two blood group system. The first is the ABO blood group system and the second is rhesus, or that is what we call RH blood grouping system. So first of all, let us come on to the ABO blood grouping system. This ABO blood grouping system was given by Karl Landsteiner in 1901. This is a very frequently asked question in all the examinations, and you must know also that this ABO blood grouping system was given by Karl Landsteiner in 1901. In the human blood, there are two antigens and two antibodies, A and B. And this ABO blood grouping system is based entirely on these two antigen and antibodies only, A and B. Antigen A and B are present on the surface of red blood cells. As I have told you earlier also that for determining any blood group type, we must know the antigen. The antigen determines that what will be the blood group of the person. So whatever antigen is present on the surface of the RBC, that will be the blood group of the person. RBCs are also known as erythrocytes. And there will be opposite antibodies present in the plasma. So that this we'll discuss later, that in case of blood transfusion how these antibodies and antigen play their roles. So for now, just keep in mind that the antigens are present on the surface of the RBC and the antibodies are present in the plasma. On the basis of the presence of the antigen, blood type is classified into mainly four groups: Group A, Group B, Group AB and Group O. So these are the four blood group types that we all know, and this is due to the presence of the specific antigen on the surface of the red blood cell. Now we are going to see that how to name the blood groups on the basis of the presence of the antigen. So first of all, here I will be discussing about the blood group A. You can all see this diagram. In this diagram, this orange portion represents the RBC, the red blood cell, and these are the antigens which are attached to the RBC, which are there on the surface of the RBC. So as you all can see that on the surface of the RBC there is the presence of A antigen, and this green portion is your plasma, and here is the presence of B antibodies. So as I told you that the blood group will be given according to the name of the antigen that is present on the surface of the RBC. So here A antigen is present on the surface of the RBC. That is why this blood group is known as blood group A. Now See this, antigen A present on the surface of the RBC and antibody B is present in the plasma. Now let me clear a very small thing here, that why the antibody B is present in the blood cell, in the plasma. Antibody B means against B. This antibody B is going to work against the antigen B, because in the blood of this person, the antigens present on the RBC are A. So whenever a antigen B will come into the blood, that will not be compatible with the blood type. So that is why antibody B is present here to protect the person against the invasion of antigen B if in case it enters the person's body. Now let's come on to the blood group B. Here in the blood group B, you can see that on the surface of the RBC, antigen B is present, and in the plasma, the antibody A is present to protect it from antigen A if in case it enters the blood. So, due to the presence of the antigen B, this blood group will be known as blood group B. Now let's come on to the blood group AB. Since the name of the blood group is AB itself, so this indicates that both antigen A and B will be present on the surface of the RBC. That is why its name is blood group AB. And when we talk about the antibodies, you cannot see here any antibody present. Why? Because both the type of antigens are present on the surface of the RBC. So if any of the antibody will be present here, then it will be affecting the antigens. It will be destroying the antigens present on the surface of RBC. So here you can see that both antigen A and B are present on the RBC, but there is no antibody present in the plasma. Let's see blood group O. See there, this O indicates 0, that there will be no antigen present on the surface of the RBC. And since no antigen is present on the surface of the RBC, therefore both A and B will be present in the plasma. So why the name O is given? Because there is absence of both the antigens on the surface of RBC, and both antibody A and B are present in the plasma. So we can summarize this whole concept in the form of this table, that blood group A will be having antigen A and antibody B. Blood group B will have antigen B and antibody A. Blood group AB will have both the antigens A and B, but there will be absence of any kind of antibody in the plasma. Then blood group O will have no antigen present, and then both the antibodies will be present in the plasma, both A and B. Now we come on to the next system, that is the rhesus system, and it is very easy to understand. The rhesus system mainly classifies all the blood groups in major two categories based on the presence of antigen D. Antigen D is to be considered in the rhesus system. So it classifies the blood groups in mainly two types. The first is positive. If antigen D is present on the surface of the RBC, then the blood group is known as positive. And if the antigen D is not present on the surface of the RBC, then this blood group is said to be negative. So you all must have heard the person is having A positive or A negative or B positive or B negative. This is because of this RH system only. A and B is given according to the ABO system, and this positive and negative is given according to the RH system. So now, considering both the systems together, we can conclude that there are total 8 blood groups: A positive, A negative, B positive, B negative, AB positive, AB negative, O positive and O negative. So these are the 8 blood groups which we have found after studying the two systems, ABO system and the RH system. So this is about the basics of the blood grouping. So this I have covered in part 1. Now the blood grouping compatibility, that which blood group can give blood, can donate the blood to which other blood group. So this you will be understanding in the part 2. And one more thing, that why why O negative is considered to be donor and why AB positive is considered to be universal recipient. All these things we'll be discussing in the part 2.